Welcome to the Deep Dive, your shortcut to understanding the complex research shaping our world. Today, we are going deep into cellular dynamics, how cells grow, move, divide, all that stuff. And specifically, how we study this inside these incredible, complex, miniature organs grown in the lab, organoid cultures.
Right, and time-lapse microscopy gives us these amazing movies of life happening inside them.
But watching is one thing, analyzing is another, and for that, AI-driven cell tracking algorithms are, well, they're essential tools. Okay, let's unpack this. Because the problem is, even though we really rely on these algorithms, they have this fundamental, pretty critical flaw. It really undermines the scientific rigor.
Yeah, exactly. And that flaw is basically the lack of statistical confidence. I mean, think about any standard biological analysis you run, like sequence alignment or gene expression analysis. You always get some kind of statistical measure, right?
Yeah.
A p-value, maybe a confidence interval.
Right. It tells you how much to trust the result.
Precisely. And that metric is foundational. It tells you, how reliable your data actually is. But cell tracking, for decades, it's been, well, kind of a statistical black box. The algorithm just spits out one single answer, one tracking solution.
With no alternatives?
No alternatives presented, and crucially, no probability attached. It just says this is the track. And if it makes just one tiny error, say it gets the timing of a cell division wrong or mixes up two sister cells, that single mistake can completely cascade and change your entire biological interpretation. Maybe it makes a symmetric division look asymmetric. Big consequences.
Okay, so it's not just about getting it right most of the time. It's about knowing how right it is.
Exactly. Without those confidence stats for every single step, every link between cells, well, reproducibility and transparency just kind of crumble.
So that brings us to what we're diving into today. OrganoidTracker 2.0, or OT 2.0. And this sounds like a real game changer because it tackles exactly that problem. It provides the cell tracks, but it also gives error probabilities for every single step, like p-values, but for cell movement and lineage.
That's the goal, bringing that statistical rigor that's been missing.
Okay, but let's back up a second. Why is this tracking so hard in the first place? I mean, hundreds of cells in 3D.
Oh, it's incredibly challenging. You've got hundreds, sometimes thousands of cells packed really densely in these 3D organoid structures. Their nuclei are often squished right up against each other, and they move fast, especially during cell division. It's chaotic visually.
And the old methods just couldn't keep up.
Not reliably. Existing methods often relied on these kind of crude, hand-tuned rules, heuristics basically, like if a cell suddenly jumps more than, say, 5 microns, flag it as a potential error. Or if the nucleus volume changes dramatically fast, maybe that's a division or an error.
Sounds a bit arbitrary.
It was. And because those rules were so basic and often wrong, it meant researchers had to manually check or curate massive amounts of data.
How massive?
We're talking potentially checking every single link the algorithm made. For just one movie of one organoid, maybe 60 hours long with a few hundred cells, that could literally take days of painstaking work.
Days. Okay, so that completely rules out doing any kind of large-scale experiment, like testing different drugs or conditions.
Absolutely impossible for high-throughput screening. Just way too slow, too much manual effort.
So how does OrganoidTracker 2.0 get around this? You mentioned two key ideas.
Right. Two main conceptual innovations. First, it uses specific modular neural networks. And these are trained not just to track, but to produce initial high-quality probability estimates for every possible link and division event.
Okay, probabilities from the start. And the second?
The second is borrowing concepts from statistical physics, things like microstates, partition functions, fancy terms. But the idea is to use these to combine those initial and end probabilities into much smarter, context-aware error probabilities. It looks at the bigger picture.
Interesting. Physics helping biology. Let's break down that first part. The neural networks. How does it even start? Detecting the cells.
Yeah, detection first. They use a 3D UNet, which is a common type of neural network for image segmentation. But they added a clever twist. Instead of just finding cell boundaries, it predicts something they call an adaptive distance map.
Adaptive distance map. What does that do?
So imagine two nuclei really close together, almost touching. Standard methods often merge them into one blob that's under segmentation, a common error. This adaptive distance map basically tells the algorithm, hey, even though these pixels are close to both cell centers, there must be a boundary here. It computationally pushes the centers apart where they are too close, creating a kind of buffer zone.
Ah, so it helps separate cells that are packed really tightly.
Exactly. It significantly reduces those undersegmentation errors, which is crucial in these dense 3D cultures.
Okay, cells detected. Now, the linking part connecting cell A in this frame to cell A prime in the next frame, this is the core tracking challenge, right?
Right. And this is where the specialized NNs come in again.
Now, wait a minute. My understanding is that while neural networks are great at classifying things, is this a cat or a dog? They're often not so good at giving reliable probability scores. Sometimes they're overconfident.
That's a really important point, and they address it directly. These specific NNs are trained and then calibrated specifically to make sure their output score really does reflect an accurate probability.
How do they do that?
They train them using 3D image crops around potential links. So the NN looks at the image data around cell A in frame 1 and cell A prime in frame 2 and predicts the likelihood that it's a genuine link, either the same cell moving or a mother cell dividing into two daughters. They use standard techniques like cross-entropy loss during training and something called Platt scaling afterwards to make sure the output numbers behave like true probabilities.
And these probabilities, they call them energy.
Yeah, it's a convenient mathematical transformation. They take the negative logarithm of the likelihood, so a high likelihood becomes a low energy. A low likelihood event gets a high energy penalty.
So low energy means this looks good, very plausible.
Exactly. Low energy equals high plausibility. It's just an intuitive way to think about the cost or penalty associated with accepting that link or division.
And does this energy approach work better than just, say, picking the closest cell in the next frame?
Oh, much better. Their results show these NNs correctly identify true links even when the cell moves quite a bit, like 3 to 7 micrometers between frames. That kind of jump happens often, especially during division.
And the simple closest neighbor approach would fail there.
Absolutely. Fails exactly when things get interesting and dynamic. The NN approach, using the actual image context, is far more robust for building accurate lineage trees. And they even have a third, separate NN just for spotting divisions accurately.
How does that one work?
It looks at image crops across 3 time points, the frame before, the frame of, and the frame after a potential division. It's trained to recognize the specific shape, the morphology of a nucleus undergoing division, like that metaphase plate structure.
It's accurate.
Very.
Yeah.
They report over 90% accuracy in pinpointing the exact moment of division, which is crucial for things like measuring cell cycle timing.
Okay, let's take a quick pause here. A mini recap. We started with the big problem. Cell tracking algorithms gave us tracks, but no sense of how reliable they were, no statistical confidence.
Right. A major bottleneck for rigorous biology.
OrganoidTracker 2.0 tackles this first by using these specialized, carefully calibrated neural networks. One detects cells using that adaptive distance map. Others predict the energy, basically, the likelihood of every possible link and division event based on the 3D image data.
Yep. So we get these initial, let's call them naive, probability estimates for every single potential step in the track.
Which leads us perfectly to the next question, maybe one you're thinking about too. If the neural network already gives us a pretty good calibrated probability score for each link, why isn't that enough? Why bring in complex statistical physics?
Yeah, that's the million-dollar question, isn't it? And this is where the really cool conceptual leap happens.
Okay, lay it on us. Why isn't the NN's local score good enough?
Because that NN prediction is, as I said, naive. It's based only on a small image crop around the potential link. It has no idea what's happening with the thousands of other potential links and cells across the entire movie. It lacks context.
Ah, context. Like knowing what happens nearby affects the probability of something specific.
Exactly. Think about how a human would do this manually. If you're tracking, you probably find the really obvious high-confidence links first, right? The cells that barely moved.
Sure, lock those in.
And once you've locked those in, they act as constraints. They immediately rule out alternative, maybe slightly confusing possibilities for the cells nearby. Establishing the easy links helps you resolve the harder ones.
Okay, that makes sense. You use the certain stuff to figure out the uncertain stuff.
Precisely. OrganoidTracker 2.0 simulates that kind of global reasoning, that common sense, using concepts from statistical physics.
How? What are these physics concepts? Microstates.
Okay, so think of every single possible way you could connect all the dots, every detected cell across all the time frames in the entire movie. Every possible complete set of tracks, including all divisions. Each one of these complete tracking solutions is like a possible story or narrative of what happened. In physics terms, each complete narrative is called a microstate.
Every possible story. That sounds like an astronomical number of possibilities.
It is. Absolutely astronomical. But stick with the concept. The likelihood or probability of any one complete narrative is related to its total energy. You just add up the link energy and division energy penalties from the NNs for every single step in that narrative.
So a narrative built from lots of low-energy, high-likelihood steps is itself very likely.
Exactly. Its overall probability is proportional to E, where E is that total energy. Low total energy means high overall probability for that specific narrative.
Okay, I follow the concept, but you can't actually calculate the energy for every possible narrative, can you? It would take infinite computing power.
Correct. You can't enumerate all microstates. So they use a clever mathematical technique called marginalization. This is the key. Instead of trying to find the single best overall narrative, which is what older methods implicitly did, they focus on the confidence of one specific link. Let's call it link A. They calculate the context-aware likelihood of link A by summing up the likelihoods, those E terms, of all the plausible narratives that happen to contain link A.
Okay, sum up all stories that include that specific link.
And then, crucially, they normalize that sum. They divide it by the total likelihood of all possible narratives, whether they contain link A or not. That denominator is analogous to the partition function in statistical physics.
Whoa. Okay, slow down. So the probability of one link depends on how likely all the stories containing it are, compared to all the stories overall.
You got it. It sounds complicated, but what it does computationally is effectively average out the influence of all the other decisions being made simultaneously across the whole organoid. It lets the context, the surrounding links, influence the probability of link A.
Does it really calculate across the whole organoid movie? That still sounds computationally intense.
Ah, good point. In practice, they make a reasonable approximation. They only compute this marginalization locally, within a small subgraph around the link in question, typically looking at links less than 3 steps away in time and space. It captures most of the relevant context without being computationally impossible.
Okay, local context, but much broader than just the link itself.
Yeah,
that makes sense. So this marginalization step refines the initial NN probability using the surrounding information.
Precisely.
But there's one more piece you mentioned, this temperature factor, T. Why do they need to introduce temperature into this? It sounds weird.
Yeah, it does sound a bit strange initially, but it's the final critical calibration step. Remember how the NNs predict the initial energies based on image props? Well, those image crops often overlap quite a bit, especially for nearby potential links. So the NNs are looking at partially shared information. Now, if you just combine those initial NN predictions using the marginalization formula, assuming they are independent, you run into a problem. It's like asking 5 friends who all saw the same slightly blurry event from slightly different angles. They might all confidently agree on an interpretation because they're all using some of the same fuzzy data.
So combining their slightly correlated reports makes you overconfident in the result.
Exactly. The marginalization process, if you use the raw NN energies directly, tends to produce probabilities that are too confident, too close to 0% or 100%. The predicted error rates don't quite match the actual error rates you find when you check against ground truth data.
So temperature T fixes this overconfidence.
That's its job. They introduce this factor T, which is greater than 1, and they essentially divide all the energy penalties by T. Dividing by T makes the energy differences less extreme. It effectively reduces the confidence derived from each individual NN prediction before they get combined in the marginalization step.
Like turning down the contrast on the probabilities.
That's a decent analogy. They tune this single temperature value, T, by comparing the algorithm's predicted error rates on a test dataset to the known true error rates. They adjust T until the predicted probabilities are perfectly calibrated, until a predicted 99% confidence actually means a 1% error rate in reality.
Okay, so T is a global calibration factor that corrects for the inherent correlations in the NN inputs, ensuring the final context-aware probabilities are statistically reliable.
You've got it. And the results show this works remarkably well. The context-aware predictions after temperature scaling show much better agreement with the actual measured likelihoods, especially for those tricky links like large displacements.
Fantastic. So we have detection, NN-based initial probabilities, context refinement via marginalization, and final calibration with temperature. Let's talk impact. What does this statistically grounded tracking mean for actual research?
Well, first off, the raw accuracy is highly competitive. They achieved error rates below 0.5% per cell per frame on challenging intestinal organoid data, and that's before any human correction.
Less than 0.5% error per cell per frame. That's impressive.
It is, and that leads directly to the first huge practical benefit. drastically reduced manual curation time.
Because you trust the probabilities now.
Exactly. Instead of checking every single link like before, which took days, researchers can now just focus on the links the algorithm itself flags as low confidence, say anything below a 99% probability threshold.
So you only review the dubious bits.
Right. And the sources note that for a typical 60-hour movie, this approach dropped the manual correction time from days down to just a few hours.
Days to hours, that's a massive difference. And you're still catching the real errors.
Mostly, yes. You might miss a tiny fraction if the algorithm was overconfident, but you're catching the vast majority of issues flagged by the low confidence score. The overall error rate after this targeted curation is extremely low.
Okay, faster curation is great, but you mentioned high-throughput screening earlier. Does this enable fully automated analysis?
Yes, and that's perhaps the biggest breakthrough for fields like drug discovery or developmental biology screens. For really large-scale experiments, you might not even do any manual correction.
How does that work? You just ignore the low-confidence parts.
Essentially, yes. The system can be set to simply remove or discard track segments that fall below your chosen confidence threshold. You're left with a collection of high-confidence track fragments.
But doesn't that leave gaps in the data? Incomplete tracks.
It does, but, and this is clever, you can then use statistical methods specifically designed to handle incomplete or censored data.
Like what?
They showcase this brilliantly using a method called survival analysis. It's a framework often used in clinical trials, for instance, to analyze patient survival times, even when some patients drop out of the study early. It's built to handle exactly this kind of incomplete data.
So they applied survival analysis to the high-confidence track fragments.
Correct. They did this for 20 different organoid movies completely automatically, using only the fragments above a high-confidence threshold.
And could they still extract meaningful biological information?
Absolutely. Using survival analysis on these automatically generated fragmented tracks, they were able to calculate key lineage features with proper statistical backing. For example, they found the average cell cycle time was around 17 hours, plus or minus about 2.8 hours. They estimated the differentiation rate, the proportion of cells that stopped dividing, at about 32%.
Wow. Automatically from fragmented data.
And they could even quantify more subtle things like sister cell symmetry. They found that 97% of sister cells divided within about 10 hours of each other. These are sophisticated biological insights derived without any manual tracking or correction, just by using the high-confidence data.
And crucially, tying it back to the original problem, the error probabilities themselves provide the statistical rigor.
Yes, they function just like the p-values we wanted from the start. If you want to know the confidence in a complex feature, like the calculated cell cycle time for one specific cell, or even the structure of an entire lineage tree, you can estimate its overall probability. It's surprisingly simple. The overall confidence is basically 1 minus the probability that any of its constituent links were wrong. Mathematically, it's p key equals 1 prada pi, where those pi are the context-aware probabilities of all the individual links making up that feature.
So you just multiply the probabilities of all the steps involved.
Essentially, yes. Multiply the individual success probabilities to get the overall success probability. This allows scientists to put a clear quantitative confidence score on their findings. Moving from, we think this happened, to, we are 99.8% confident this lineage tree is correct up to this point.
That's the transparency and rigor that was missing. What about versatility? Does this only work on intestinal organoids?
No, and that's another strength. They tested OrganoidTracker 2.0 on completely different data sets, mouse blastocysts, even C. elegans embryos, which very different and were imaged under different conditions.
And it worked. Did they have to retrain the neural networks completely?
It performed well on these out-of-sample data sets. And interestingly, they often found they didn't need to do a full, costly retraining of the complex neural networks. Frequently, just recalibrating that single temperature parameter, T, was enough to adapt the system to the new imaging conditions and maintain well-calibrated probabilities.
That's a huge practical advantage over typical machine learning workflows, where retraining can be a major bottleneck.
Definitely. It suggests the core NN features learned are quite general, and the main adaptation needed is just adjusting that final confidence calibration.
Okay, so to wrap up, OrganoidTracker 2.0 seems to represent a fundamental shift. It takes cell tracking from this kind of opaque, heuristic-driven process.
Where you just crossed your fingers and hoped the output was right.
Yeah, to a statistically rigorous, transparent scientific method. It's not just giving you the track, it's giving you the confidence score, the p-value, for that track, which was the critical missing piece.
Exactly, and the key innovation seems to be that combination. The modular NNs providing reliable initial probability estimates, coupled with that statistical physics framework, the marginalization providing the essential context and refinement.
Which allows researchers to actually assess the certainty of the claims they're making based on observing cell dynamics.
Right. And this really opens the door to reliable, high-throughput screening of cellular behaviors in complex 3D systems like organoids. That has huge implications for, well, for cancer research, developmental biology, drug discovery, you name it.
It really sounds like it could accelerate research in those areas significantly.
I think so.
So that brings us to our final thought, something for you, our listeners, to mull over. Now that researchers have a tool like OrganoidTracker 2.0 that can quantify the statistical confidence in every single piece of cell tracking data.
What previously hidden biological insights might be waiting to be discovered? Think about all those archived microscopy movies, maybe from years ago, where subtle effects were seen but couldn't be published because the tracking errors were unknown or too high to justify the claims.
Right. Could revisiting that old data with this new statistical rigor unlock discoveries that were previously just beyond our grasp due to uncertainty? What might be hiding in plain sight, just waiting for a confidence score?